The first team is Jeremy Appelbaum, William Gill, Alan Shin. They are from the George Hewlett High School in Hewlett, New York. Please welcome with me the first presenters.
Thank you. Good afternoon. My name is Jeremy Appelbaum. I'm here with Alan Shin and William Gill, and this is the regulation of scotomorphogenesis by COP1 in Ceratopteris richardii. The degradation of proteins is very essential for the regulation and maintenance of homeostasis. One way cells regulate protein degradation is through the highly conserved ubiquitin proteasome pathway. In this pathway, proteins are first targeted for degradation through the addition of highly conserved signaling proteins called ubiquitin. Ubiquitin is added onto these proteins through a series of enzymes. One group of such enzymes is the E3 ubiquitin ligases. The addition of sufficient ubiquitin onto these target proteins by these E3 ligases then signals for degradation through the appropriate proteasome. The inability to properly regulate protein degradation is implicated in a wide variety of diseases, including cancer. For instance, the deregulation of the C-gene oncoprotein may result in tumorigenesis. One factor responsible for maintaining levels of C-gene is the E3 ubiquitin ligase constitutively photomorphogenic 1, or COP1. Although COP1 has been receiving a large amount of attention due to a recent link in cancer, COP1 was first discovered over 20 years ago in plants. In plants, COP1 has pleiotropic functions, and a complete homozygous knockout has been proven to be lethal. COP1 regulates processes such as photomorphogenesis, scotomorphogenesis, circadian rhythm, and flowering. A simplified mechanism of COP1 regulation of plant growth in the dark can be seen through its degradation of hypocaudal elongated 5, or Hy5. Hy5 is necessary for the transcription of many light-induced genes. In the dark, COP1 is localized in the nucleus, where it represses photomorphogenesis through its interactions with Hy5. However, upon exposure to light, COP1 is excluded out of the nucleus and localized in the cytoplasm, thus removing its ability to interact with Hy5.
Nuclear exclusion of COP1 is an effective method of regulating COP1 interaction with various transcription factors. However, this process is very slow, requiring at least 24 hours. Upon light induction, an increase in the concentration of transcription factor Hy5 is observed within the hour. This suggests a faster mechanism of COP1 inactivation. However, this inactivation is poorly understood at a molecular and cellular level. Ceratopteris richardii is a model organism that can address this problem. It is a recently evolved polypod fern that can be used to study a variety of biological phenomena such as photomorphogenesis. The advantage of using this organism stems from its unique development and life cycle. Ceratopteris can exist independently as either a haploid gametophyte or diploid sporophyte. This biphasic life cycle allows for the observational and experimental manipulation at the cellular and whole organism level without artificial manipulation of the life cycle. Since spores that are produced are genetically identical and can be generated in large quantities, a quick isolation of a mutation is possible. And since spores are only consistent of one cell, a mutation will be propagated to all the cells of the gametophyte, and a mutation can be identified whether it is recessive or dominant in nature. And since ferns represent the divergence point in the vascular plant phylogenetic tree, Ceratopteris combines features of both higher and lower plant systems.
Recently, it has been demonstrated that the incubation of sterilized dry spores with sequence-specific double-stranded RNA can reduce the expression of targeted genes. Due to the availability of ESC libraries, the generation of this double-stranded RNA is extremely easy. The convenience of this RNA interference approach has greatly increased the value and utility of studying this process of photomorphogenesis within Ceratopteris richardii. The etiolated phenotypes of Ceratopteris richardii exhibit elongated prothalli, an increase in cell elongation, and a decrease in cell division. This is similar to that of Arabidopsis thaliana, which exhibits an elongated hypocaudal, an increase in cell elongation, and a decrease in chlorophyll accumulation.
Similar to the seeds of Arabidopsis thaliana, the germination of spores of Ceratopteris richardii is also light-regulated. Spores grown in the dark do not germinate and require red light induction. This red light induction of germination can be photoreversed by subsequent far light exposure, implicating the regulation of germination by phytochrome photoreceptors. Unique to Ceratopteris and other ferns is the blue light inhibition of germination. This phenomenon is not seen in the seeds of higher plants. It is believed that the blue light cryptochrome photoreceptor may have a novel role in these plants. In Ceratopteris, 2 mutants have been isolated that are similar to the COP-D fusca mutants that were described in Arabidopsis thaliana. These mutants were termed germ3 and germ4 and displayed de-etiolated phenotypes when grown in the dark, suggesting that the genes involved in scotomorphogenesis, such as COP1, may also be conserved in Ceratopteris. The purpose of this study was to isolate and characterize COP1 in Ceratopteris richardii. To identify and isolate COP1 in Ceratopteris, we designed multiple sets of primers to highly conserved and unique regions of COP1. We compared the COP1 sequences from a wide variety of plants such as Arabidopsis thaliana and Angiosperm, Fiscomitrella patens, a moss, and Selaginella millendorfi lycophyte to identify these regions. These regions were then blasted to ensure they were specific to COP1 and not to other genes. A fragment corresponding to approximately 800 base pairs was amplified using polymerase chain reaction and then purified, sequenced, and analyzed. The PCR product contained a continuous open reading frame and a translatable protein comprised of 258 amino acids. The translatable protein was then blasted to other sequences and showed to have an 80% homology to the COP1 of higher plants and a 55% homology to the COP1 in humans.
To determine the evolutionary relationship of COP1 within Ceratopteris to that of other COP1 sequences in other organisms, phylogenetic trees were constructed. The COP1 sequences of various organisms were aligned and formatted using bioedit, and using mega, phylogenetic trees were constructed. As you can see, Ceratopteris occupies a separate clade from seeded plants and animals. This divergence suggests that the COP1 in Ceratopteris has novel roles and possibly novel interacting factors within Ceratopteris. However, even though that there's a divergence, COP1 as an E3 ubiquitin ligase is conserved.
Structurally, COP1 possesses a zinc-binding ring finger domain, a coiled coil region, WD40 repeats, a high-5 interacting motif, and various COP1 interacting protein domains. These motifs are necessary for the function of COP1 and its regulation of scotomorphogenesis. However, the structure of COP1 in lower plants remains uncharacterized. To determine if these motifs are conserved in lower plants, we isolated uncharacterized sequences from EST libraries of Fiscomitrella and Selaginella. One of the reasons we needed these sequences is because we did not obtain the full-length COP1 sequence from Ceratopteris richardii. As seen in the figure, the isolated fragment from Ceratopteris corresponds to the highly conserved WD40 domain of Arabidopsis. WD40 repeats, which are repeating tryptophan and aspartic acid residues every 40 amino acids and necessary for protein-to-protein interaction, were highly conserved among all three plants. Ceratopteris, Fiscomitrella, and Selaginella had an 83, 82, and 80% homology to the WD40 domain of Arabidopsis, respectively.
The high-5 interacting motif of Ceratopteris, Fiscomitrella, and Selaginella all had an 83.3% homology to that of Arabidopsis thaliana. This suggests that an ortholog of high-5 exists within Ceratopteris. To test this hypothesis, we scanned the EST library of Ceratopteris using blast analysis. However, the EST library is incomplete and does not represent the entire genome of Ceratopteris. We did find an ortholog of high-5 within Selaginella with a 55% homology to that of Arabidopsis thaliana. Since an ortholog of high-5 was found within Fiscomitrella and Selaginella, it is suggested that an ortholog of high-5 exists within Ceratopteris as well.
Although we only isolated a partial fragment of COP1 within Ceratopteris richardii, the ring finger domain is probably conserved as well. The ring finger domain is necessary for the recruitment of E2 ubiquitin conjugating enzymes and E2 variant proteins. The Fiscomitrella and the Selaginella sequences had a 77 and a 75% homology, respectively. The coil region is necessary for a dimerization of COP1 and its function in vivo. In order for COP1 to function, COP1 must first form a tetramer consisting of 2 COP1s and 2 SPA1s, or suppressor of phytochrome A1s. The coil region also contains the binding sites of KIP1 and KIP7, or COP1 interacting protein 1 and 7. The coil region, the Fiscomitrella and Selaginella sequences had a 59 and a 54% homology, respectively. The divergence of this region suggests a greater variability of binding factors within Ceratopteris richardii. Analysis of the EST library revealed a putative ortholog of SPA1 with a 40% identity. The nucleolocalizational signal, or the NLS, is responsible for the localization of COP1 within the nucleus. The NLS is extremely well conserved in both Fiscomitrella and Selaginella with 100% identity in both. This would suggest that the NLS is also conserved in Ceratopteris richardii as well. To determine the function of COP1 within Ceratopteris richardii, sterilized dry spores were incubated with double-stranded RNA for 48 hours and then exposed to light for 3 days to induce germination. These spores were then plated and placed in the dark for 7 days. The spores treated with double-stranded RNA exhibited a shortened prothalli and a decrease in cell elongation. These prothalli, however, contain more cells in the prothalli of gametophytes that are not treated with double-stranded RNA. The phenotypes we observed were similar to that of the germ3 and germ4 mutants previously characterized.
Previous physiological studies have shown that cytokinin can induce limited photomorphogenic properties in the dark. Cell elongation decreased while cell division increased in spores treated with kinetin, a cytokinin. To determine if cytokinin affects the transcription of COP1, we performed an RT-PCR. Spores that were grown in the dark and treated with kinetin showed no change in the relative expression of COP1 as compared to the control, which had no kinetin. To ensure that equal levels of cDNA template were used, we amplified the ubiquitin gene as a control because it is usually constitutively expressed and at a relatively constant level. However, these results are not absolutely quantitative, but rather semi-quantitative. We also amplified these genes during the mid-log phase to ensure we could see a difference in the expression.
In conclusion, we isolated a partial fragment of the ortholog COP1 gene within Ceratopteris richardii.
This fragment contains essential WD-40 motifs and necessary for COP1 interactions with other proteins.
Lastly, COP1 in lower plants retains its primordial role to regulate the etiolation of gametophytes.
For future research, we would like to isolate and characterize the entire COP1 sequence within Ceratopteris richardii using raised RT-PCR. Furthermore, we would like to understand its localization using fusion fluorescent proteins and cytochemistry studies. We would also like to use the COP1 gene to look at possible novel interacting factors and high-fi phytochromes and cryptochromes within Ceratopteris using the yeast-2 hybrid system. We would also like to isolate and characterize the COP1 genes within the mutants germ3, germ4, and the DKG1 mutants. And we would also like to look into the blue light inhibition of germination within Ceratopteris richardii.
We'd like to thank everyone at Siemens and College Board and all the judges here today who dedicated their time to allow this event to happen. We'd also like to thank you for inspiring students in the world of STEM. And lastly, we'd like to thank our mentor, Dr. Terrence Bissoondial, for introducing us to these photobiological processes and techniques that we used. Thank you.